Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Apple scab disease, causes, symptoms, and best prevention tips for apple and crabapple trees. Table of Contents. Introduction. Causal organism. Symptoms. Morphology. Reproduction. Apple scab disease cycle and epidemiology. Environmental factors impacting epidemiology. Apple scab disease management. Introduction to apple scab disease. Apple scab disease is among the most devastating diseases impacting apple and crabapple trees, significantly reducing yield, fruit quality, and overall economic value. First identified in the 19th century, it has since posed a persistent threat to apple orchards. The disease is prevalent in temperate climatic regions, where the pathogen thrives under cool and moist environmental conditions. Causal organism of apple scab disease. Causal organism. Apple scab disease is caused by the fungus Venturia inaequalius, which belongs to the family Venturiaceae, order Pleosporales, class Dothodiomycetes, and phylum Ascomycota. It is a hemibiotrophic pathogen, meaning it does not directly kill the host, but instead depends on it for nutrition. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Symptoms of apple scab disease. Symptoms on leaves. The disease first appears as small olive green to dark brown spots on the leaves, which are generally circular, highly raised, and have a velvety texture. These spots typically develop on the underside of young leaves, as they are more susceptible to infection. As the disease progresses, the spots increase in size and may merge to form large blotches. The color of the lesions darkens over time, turning deep brown or almost black in severe infections. Infected leaves may become twisted or deformed as the fungal infection interferes with normal growth patterns. The deformation results from abnormal fungal growth, causing irregular expansion of leaf tissues. In severe cases, the infection can lead to defoliation, where leaves prematurely drop from the tree. Defoliation weakens the tree, reducing its ability to photosynthesize and negatively affecting overall health and productivity. Symptoms on fruits. The initial symptoms on fruits resemble those on leaves, appearing as small olive green to dark brown spots. As the fruit matures, these spots develop into more prominent scabs, which are rough in texture. The infected areas become harsh and corky, giving the fruit a characteristic scabbed appearance. The scabs may appear as isolated patches or expand to cover a large portion of the fruit's surface. In severe infections, the fruit may crack as it continues to grow, leading to structural damage. These cracks create entry points for secondary pathogens, increasing the risk of further infections and fruit rot. Twigs and blossom symptoms. Apple scab can also infect tree twigs, causing lesions similar to those on leaves and fruits. The lesions on twigs may appear elongated rather than circular. Blossoms are also susceptible to infection, developing small, dark lesions similar to those found on leaves. Infected blossoms may wither and drop prematurely, reducing the number of developing fruits. Severe infections can significantly impact fruit production, leading to economic losses in apple orchards. Morphology of the host. Venturia inaequalis. The mycelium of Venturia inaequalis is septate, meaning it is divided by cross walls. The hyphae grow beneath the epidermal layer of the host tissue and contain haploid nuclei. Under favorable conditions, the fungus produces both sexual and asexual spores. A dark brown ring-like structure, known as the melanized apressorial ring, forms at the base of the apressoria. During sexual reproduction, it develops fruiting bodies, called pseudothecia, which facilitate the production of sexual spores. Reproduction in Venturia inaequalis. Asexual reproduction. Venturia inaequalis reproduces asexually through the formation of conidia. Conidia are unicellular spores, typically brown to olive green, and are produced on conidiophores. Conidiophores can be short-branched or unbranched hyphae, appearing hyaline to pale brown in color. The surface of conidia is sticky, aiding in their adhesion to fruits and leaves, which facilitates secondary infection. Sexual reproduction. Venturia inaequalis reproduces sexually by forming fruiting bodies known as pseudothecia. During sexual reproduction, an antheridium develops from the hyphal tip of one individual, while a trichogyne forms from the hyphal tip of another opposite individual. Fertilization occurs when the nucleus from the antheridium is transferred to the trichogyne and incorporated into a cell at the base of the pseudothecial initial. After fertilization, the pseudothecial initial matures into a pseudothecium, which develops inside a cavity known as a stroma. Pseudothecia are flask-shaped structures that become deeply embedded in dead leaf tissue. The walls of the pseudothecia are composed of melanin-rich cells, giving them a dark brown to black appearance. The diploid nucleus inside the pseudothecia undergoes meiosis, forming 8 haploid ascospores within a sac-like structure called azae. Each ascospore is ellipsoid to fusiform in shape and consists of 2 cells separated by a central septum. Ascospores are released through a small opening in the pseudothecia called the osteole. During moist conditions, pseudothecia protrude and detach from leaves, and ascospores are dispersed by rain and wind. Ascospores serve as the primary inoculum, initiating primary infection in the next growing season. Apple scab disease cycle and epidemiology. Primary infection phase. The primary infection phase of apple scab disease begins in early spring with the release of ascospores from pseudothecia. Ascospore release. As apple leaves start emerging in early spring, pseudothecia from the previous season absorb moisture from rain or dew. This moisture causes pseudothecia to swell, leading to the discharge of ascospores through the osteole. Since moisture is crucial for both spore discharge and infection, the synchronization of wet periods plays a vital role in disease progression. Dispersal and germination. Released ascospores are carried by wind to newly emerging leaves and flower buds. For germination, these susceptible tissues must have sufficient surface moisture. Once settled, the ascospore germinates, forming a germ tube that penetrates the epidermis and establishes the fungal mycelium beneath the surface. Primary lesion formation. The first visible signs of apple scab appear as primary lesions on young leaves or fruits, resulting from the initial ascospore infection. These infections begin as small olive green spots, which gradually darken and develop into smooth scabs as they mature. Secondary infection phase. Venturia inaequalis progresses into the secondary infection phase following the establishment of the primary infection. Conidia play a crucial role in spreading the disease during this phase, leading to secondary infections. Conidia production and dissemination. Conidia are formed on conidiophores as a result of asexual reproduction occurring on primary lesions. These conidia are frequently released and transmitted via wind and rain, causing new infections on different parts of the same tree or nearby trees. Infection spread. Conidia are primarily spread by the rain splash and wind, leading to new waves of infection. Secondary infections tend to be more severe than primary infections since they can occur repeatedly and spread rapidly. Lesion formation. Secondary infections lead to new lesions on leaves and fruits. These secondary lesions serve as fresh sources of conidia, further fueling the disease cycle and accelerating its spread. Advancement of disease. Under favorable environmental conditions, the combined effect of primary and secondary infections throughout the growing season can result in multiple outbreaks. Leaf senescence and defoliation. Severe infections can cause early leaf senescence and defoliation, significantly reducing photosynthetic capacity. This can weaken the tree and negatively impact fruit production. Damage to the fruit. Infected fruits develop corky, dark lesions that frequently crack as the fruit grows. Such damaged fruits are unmarketable, leading to economic losses due to reduced yield and fruit quality. Overwintering phase. As the growing season comes to an end, Venturia Inaequalis transitions into its overwintering phase, setting the stage for the next infection cycle. Pseudothecia formation and maturation. During autumn, infected leaves fall to the ground, providing a substrate for pseudothecia formation. The mycelium undergoes sexual reproduction during winter, leading to the maturation of pseudothecia. These mature pseudothecia will eventually release ascospores in early spring, initiating a new cycle of infection once favorable conditions return. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Environmental factors impacting epidemiology. Temperature. The optimal temperature range for apple scab development is 12 degrees Celsius to 24 degrees Celsius, 54 degrees Fahrenheit to 75 degrees Fahrenheit. At temperatures below 5 degrees Celsius, ascospore release is inhibited, reducing the risk of infection. At temperatures above 25 degrees Celsius, the pathogen's infectivity declines significantly, limiting disease progression. Moisture. Moisture plays a crucial role in ascospore release and conidia germination. Prolonged leaf wetness caused by rain, dew, or irrigation is essential for infection. At temperatures between 12 degrees Celsius and 20 degrees Celsius, even 6 hours of leaf wetness can result in significant infection levels. Humidity. High relative humidity, above 90%, favors conidia production and dissemination during the growing season. Prolonged high humidity significantly increases infection pressure, leading to severe outbreaks. Host susceptibility. The susceptibility of apple cultivars influences disease severity. Resistant cultivars include Macintosh and Granny Smith, while highly susceptible cultivars include Liberty and Enterprise. However, even resistant cultivars can be affected under highly favorable conditions, or due to the emergence of virulent pathogen strains. Pathogen variability and virulence. Venturia inaequalius can mutate and evolve into new races capable of overcoming resistance genes in apple cultivars. This evolutionary adaptability necessitates continuous breeding of resistant varieties and implementation of disease management strategies to control outbreaks. Apple scab disease management. Effective management of apple scab requires a combination of cultural practices, resistant cultivars, fungicide applications, monitoring, and biological control strategies. First, cultural practices. Implementing proper orchard management reduces the survival and spread of Venturia inaequalis. Sanitation. Fallen leaves and debris harbor overwintering pseudothecia, serving as a primary inoculum source. Removing or mulching infected leaves significantly reduces early spring infections. Pruning. Proper pruning improves airflow within the tree canopy, reducing leaf wetness duration. Thinning dense foliage allows leaves and fruits to dry faster, making it harder for the pathogen to establish. Diversification. Planting a mix of resistant and susceptible cultivars slows disease spread, as resistant trees act as barriers to pathogen transmission. Second, resistant cultivars. Breeding for resistance has led to the development of apple varieties with improved defenses against Venturia inaequalis. Cultivars such as Liberty, Enterprise, and Gold Rush exhibit strong resistance. However, resistance is not absolute, as the pathogen can mutate over time. Continuous breeding efforts are essential. Third, fungicide applications. Fungicides are essential in managing apple scab, particularly in orchards with susceptible cultivars or where environmental conditions favor infection. Timing. Fungicides are most effective when applied preventatively before infection occurs. Critical application periods include pre-bud break, early leaf expansion, integrated fungicide programs. Rotating fungicides with different modes of action prevents pathogen resistance development. Applying fungicides at the lowest effective dose and following recommended intervals maintains long-term efficacy. Organic options. Sulfur and copper-based fungicides are commonly used in organic orchards. While less effective than synthetic fungicides, they provide moderate control when combined with cultural practices and resistant cultivars. Fourth, monitoring and forecasting. Regular scouting and predictive models help in early disease detection and timely interventions. Scouting. Frequent orchard inspections identify early apple scab symptoms. Detecting initial lesions helps prevent disease escalation and optimizes fungicide use. Weather monitoring. Disease outbreaks are closely tied to temperature, humidity, and rainfall. Tracking weather conditions helps predict spore release and infection periods. Disease prediction models. These models use weather data to assess infection risk and optimize fungicide application timing. They help reduce costs and minimize environmental impact by preventing unnecessary treatments. Fifth, biological control. Using natural enemies or antagonistic microorganisms can help suppress Venturia inaequalis. Antagonistic microorganisms, beneficial fungi and bacteria, are being studied for their ability to outcompete or inhibit the pathogen. While still in research phases, biological control offers a sustainable alternative for apple scab management. Compost teas. These microbial-rich sprays, derived from composted organic matter, may suppress apple scab pathogens, an eco-friendly and cost-effective option for disease management. Sixth, post-harvest management. Apple scab control efforts should continue beyond harvest to reduce pathogen carryover. Leaf litter management. Removing or mulching infected leaves disrupts the pathogen's overwintering cycle. Orchard floor management. Keeping the orchard floor clean reduces pathogen survival. Cover cropping or mulching promotes beneficial microorganisms that decompose infected material. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!